All right, welcome everyone to our SVP Cancer Center seminar series with our new Focus On format that features two excellent speakers. Today's presentations will focus on the tumor microenvironment. Each presentation will be followed by a Q&A. So please use the Q&A function within Zoom to submit your questions. There will also be time at the end for additional discussion and follow-up questions. So our first speaker is Dr. Madeleine Oudin, who joins us from Massachusetts today. Madeleine graduated from McGill University prior to completing her Ph.D. in neuroscience at King's College London. Completed her postdoctoral training at MIT in Frank Gertler's lab, where her research was focused on understanding metastasis and mechanisms of chemotherapeutic resistance. She started as an assistant professor at Tufts University in the Department of Biomedical Engineering in 2018, where her lab is focused on understanding the mechanisms which the tumor microenvironment contributes to cancer metastasis and resistance to drugs. She was awarded a Young Investigator Award from the Breast Alliance and currently holds an R00 award from the NCI. Thank you, and we welcome Dr. Madeleine Oudin.
Hi, everyone, and hi, thank you so much for the introduction, and thanks for the invitation to be here today to share some of our work, along with work from Marina Pasca's lab. So we're focusing today on the microenvironment and its role in treatment resistance. And so, my lab focuses on breast cancer. We focus particularly on triple-negative breast cancer, a subtype that affects about 15 to 20% of breast cancer patients, and is characterized by the lack of expression of three main receptors that can be used and leveraged for treatment in the other subtypes. But due to the absence of these receptors, these patients have limited treatment options. There's been a few newer treatments approved recently, but still, this is a really hard disease to treat. About 30% of patients metastasize to a range of organs in the brain, bone, lung, and liver. And we know that metastasis is largely the leading cause of death in these patients. And once the cancer has disseminated, it's extremely hard to treat. We study this process really from the lens of cell migration and how cell migration is important for all these steps of dissemination. Cell migration has been shown to be important even for tumor growth, and tumors that can proliferate as well as migrate tend to grow bigger and larger. Cells will then locally invade in their surrounding tissues to reach either the vasculature or the lymphatics to disseminate. And then once they reach secondary organs, cell migration is also important in the growth of these tumors. And that tumors in these secondary sites that are able to migrate locally will form larger tumors in these secondary sites. And inhibiting migration can inhibit the number of secondary metastases form. So cell migration is really important for all steps of the metastatic cascade. Of course, cells are not alone in their environment, and the tumor microenvironment plays a really important role in controlling metastasis and dissemination. And shown here is a very reductionist schematic of the tumor microenvironment, which we know is largely composed of healthy cells and our tumor cells growing and interacting with them, a range of stromal cells, which we'll hear a lot more from in the second talk. And I will today focus on the extracellular matrix, the protein scaffolds that provide structure and support to our tissues. So the extracellular matrix is a major component of the tumor microenvironment. And in the last 5 or so years, we've been really able to characterize the composition of this extracellular matrix using proteomics. And pioneering work from Richard Heinz's lab and Alexandra Naba has led to the characterization of the matrosome, or the proteins that make up the fraction, this non-cellular protein fraction. And the core matrosome is made up of glycoproteins, collagens, and proteoglycans. And we have about 270 of these different matrix proteins, as well as a host of other matrosome-associated proteins that are consistently enriched in this non-cellular fraction that make up are composed of ECM regulators like crosslinkers and proteases, secreted factors, as well as other affiliated factors. So we now have a good understanding of the proteins present in this environment. Tumor progression is also associated with major changes in the extracellular matrix that promote many properties, including invasion. And these are summarized here. This is a really nice recent review written by Thomas Cox that just came out, summarizing all the changes in the matrix that can occur as tumors form, such as degradation and turnover, change in composition, change in cross-linking leading to changes in biomechanical properties, and all of these can contribute to progression. I'll give you just a couple of examples for breast cancer. Breast tissue is really rich in collagen 1, and these curly coiled collagen fibers can get reorganized into straight aligned fibers in the breast tissue. This is work from Patty Keeley's lab. And it's been shown that a higher proportion of these straight aligned fibers is associated with more invasive disease, and that these can provide fibers for cells to migrate on and invade locally. We can also have very heterogeneous distribution of matrix proteins and the formation of endogenous gradients of matrix proteins that can promote local invasion. So in my postdoc, I worked on fibronectin, another abundant matrix protein in breast tissue, which in the tumor tissue is highly abundant around the blood vessels shown here. And so you have these endogenous gradients that lead to tumor cells being attracted to the blood vessels and helping them disseminate. So my lab tries to understand, how these cells integrate, tumor cells integrate cues from the extracellular matrix that allow them to locally invade and metastasize, but also then how do current treatments that are used for breast cancer, and in particular here for triple negative, impact the matrix, the response of the cells to the matrix, and ultimately metastasis. So we know that for triple negative breast cancer patients, once they get diagnosed, they're usually treated with neoadjuvant chemotherapy, which is traditionally made up of 3 main drugs: doxorubicin and cyclophosphamide, or doxorubicin also called adriamycin. This is called the ACT regimen, followed by paclitaxel. Those are the 3 main drugs that are used for these patients. Cisplatin is sometimes used, but not as commonly. So these are treated before surgery, and about 50% of these patients will display a residual cancer burden after this treatment. They'll then undergo surgery to remove any leftover tumor, but about 25% of patients exhibit recurrence within 5 years. And so clinical data has shown that this neoadjuvant chemotherapy treatment does not actually reduce the risk of local or distant recurrence in triple negative. And so what does it mean to have these cells left over here? And what is actually this treatment doing to the breast tissue? And could this be perhaps in some way impacting outcome in patients? So what defines a tumor that is resistant to chemotherapy in triple negative breast cancer? There's been a range of studies on this, and here I'm just going to focus on a few that have looked at gene expression changes in tumor cells of triple negative breast cancer patients after chemotherapy. And interestingly, tumors that have low proliferative signatures or that are more migratory are more resistant. And cell migration gene expression signatures have also been shown to be upregulated in chemoresistant triple negative breast cancer. So as a postdoc, I was interested in trying to understand the relationship between effects of drugs on proliferation and tumor size, which we know is the main measured outcome within patients, and their effects on migration. And so I took mice that I injected with human triple negative breast cancer cells into the mammary fat pad to form tumors, waited till the tumors had reached a size, a good size that they had already had metastases, and treated them with either Taxol or doxorubicin. Again, these are kind of the 2 of the most common drugs used to treat triple negative breast cancer. Taxol works by stabilizing microtubules to prevent cell division, whereas doxorubicin intercalates in the DNA to induce damage that cannot be repaired. And when we looked at the effect on tumor size at the macroscopic level, so this is what would be done in the clinic, you can see that the control tumors continue to grow, but the chemotherapy treated ones did slow down their growth. So these drugs were doing what we expected them to do. They were slowing down the growth of the tumors. However, when we looked at cell migration, and we did this by intravital imaging live in the tumors in an endpoint setting, we could see that actually only Taxol was inhibiting cell migration in the tumor, whereas doxorubicin was not. And so this shows that tumor cells that are that chemotherapy drugs have different effects on proliferation and migration, and that these drugs are great at inhibiting proliferation, but they don't always do the same when it comes to migration. And there can be tumor intrinsic mechanisms of resistance, and this is one that we uncovered in cells that can, particularly as it relates to migration. But the other thing that came up in some of these data sets was an enrichment in ECM remodeling genes. And so we were interested in trying to dissect the relationship between drug response and the ECM. Of course, it's well known that tumor cells within a tumor are more resistant to, that you need higher doses to kill cells when you give them in vivo as opposed to what you do in vitro. And largely because you're injecting in an organ, in a physiological organism, but also it's been suggested that perhaps the matrix might also play a role. And so we did simple dose response experiments looking at all 4 drugs that I mentioned earlier that have been used for triple negative. And we treated cells in 2D, and we plated them on different matrices. These are the most abundant matrix proteins, again, in breast tissue: collagen 1, fibronectin, and matrigel, which is a mix of collagen 4 and laminin. And you can see that overall, the response relative to plastic, there are actually no significant differences. Some of the matrix proteins made cells a bit more sensitive, but it was nothing significant. We then placed these cells in 3D versions of these matrices. We used a spheroid model where we embedded cells in the matrix and let the cells grow over a 4-day period and looked at the area. And what you can see is that here, particularly in response to paclitaxel, we saw significant resistance when cells were plated in fibronectin or matrigel, again, composed of laminin and collagen 4. And so this shows that the matrix does, in fact, impact response to chemotherapy in 3D versus in 2D where it doesn't. So the matrix is really an important component in this response to the drug. The other thing to keep in mind when it comes to the environment is that chemotherapy can change the environment. So we know that once tumors have been treated with chemotherapy, you get the recruitment of a range of different cells due to this inflammatory state that the tumors are in. These are summarized here. And a lot of these changes can actually be pro-tumorigenic. And importantly, all of these cells can actually secrete matrix as well. And we know that matrix deposition is also often associated or happening after inflammation. And so because many of these cells can secrete and remodel ECM, we thought it would be interesting to try and understand whether chemotherapy affects the matrix in the environment and whether perhaps changes induced by chemotherapy in the matrix could explain why some patients do recur after treatment. So how do we study responses to the matrix? As I showed you earlier, we can plate cells on recombinant proteins that are commercially available. This is great for mechanistic analyses. However, you're really only focusing on a single or defined combination of matrix. And we know that, like the proteomics studies I showed you initially, we have huge diversity of matrix proteins within our tissues. Another approach that is used are cell-derived matrices. And so this is often done using fibroblasts, which are major contributors of matrix in our tissues. And you seed them in a dish, you let them secrete matrix for 2 weeks or so, remove the cells, and then you have a more diverse, complex matrix. However, with this method, you only get matrix derived from a single cell type. And the proteomic studies of the ECM, which were done, some of them in mouse hosts with human tumor cells, really suggest that you get matrix deposition coming, or matrix secretion coming from both the tumor and the stromal compartment, and then it depends on the protein. So this is still a very biased view. And then another approach commonly used by engineers is using tissue decellularization, where you take a native tissue from in vivo, you apply detergent to remove the cells, and digest it in various ways to treat cells with it in some way. And so here you get the ECM from in vivo, but you lose a lot of the structural information when you digest it. So we kind of pulled these together and developed a novel method to study whole tissue ECM effects. This was led by Andrew, my first lab member, and Sydney, a grad student. And so we initially took various breast cancer models, a xenograft model or the genetic PYMT-MMTV mouse model. We isolated the tumors or the mammary glands, put them in detergent to remove all the cells. And so this is what the tumor will look like after, once you've removed the cells. And we then replated cells on these tissues isolated from these different tumor models and evaluated effects on cell proliferation and migration by live imaging. We verified using Western blot that we were able to remove all the cellular components by staining for these different proteins and could see indeed an enrichment in the matrix. We also verified this by H&E staining to show that we were indeed able to remove in the after decellularization tissues all the nuclear cell, the cells, the nuclei, and enrich for matrix as shown here by Picrocereus red staining. So we then seeded our cells on mammary glands obtained from naive mice that didn't have any tumors, or tumors either obtained again from the xenograft model or from the genetic model. And what you can see from these movies and these representative Rose plots is that cells that were plated on decelled ECM from tumors proliferate more and migrate a lot more than those from the native healthy mammary gland. And so this showed really for the first time that whole tissue changes induced by the presence of a tumor can promote invasion and migration and proliferation overall on the cells that are present here. So we then wanted to apply this to our chemotherapy treatment and see, does chemotherapy also induce changes in the matrix that could perhaps be having effects on the cells that are left behind after treatment? So this is a project led by a grad student in my lab, Jackson Fothery, in collaboration with another grad student, Justine. So here we took the immune competent immune competent genetic model, PYMT-MMTV, treated them again with either paclitaxel or doxorubicin. And in this model, these mice develop a lot of tumors in all mammary glands, but we were able to see that we could again slow down tumor growth. So again, showing that these are doses at which these tumors are responding and the drugs are, having an effect. At this dose, though, there was no effect on lung metastases yet. So we isolated the matrix from these different chemotherapy treated mice, and we again reseeded the cells on these matrices. And the data is shown here. And what you can see here is a representative movie and some representative Rose plots. And what you can see is that treatment with both paclitaxel or doxorubicin, the matrix that resulted in these tumors significantly increased the invasion of our tumor cells compared to vehicle-treated matrix. And so this suggests that the treatment of chemotherapy in these tumors alters the matrix in such a way that it promotes its effects on invasion. So we then performed ECM proteomics to try and identify the changes in the matrix composition of these tissues. And here you can see some volcano plots of the data and some of the matrix proteins that were changing within these tissues. And so we followed up on a few of them and ended up focusing in on collagen IV. So we validated that collagen IV was indeed upregulated or more abundant in paclitaxel or doxorubicin treated tumors. This is through an independent set of mice. So you can see in the vehicle, the collagen IV levels are much lower than in either the paclitaxel or the doxorubicin treated ones. And so this confirmed that chemotherapy treatment does indeed induce more collagen IV. We then looked in human data sets to try and see if expression of collagen IV could impact survival in the context of chemotherapy treatment. So, a lot of caveats with this, especially when it comes to the matrix, because this is based on gene expression and not protein level but we worked with what we had in terms of this. And what you can see is that in patients that were not chemotherapy treated, there was no difference in the outcome when it comes to patients with either high or low collagen 4. However, in patients that were chemotherapy treated, and this is, all the drugs, that I mentioned before, patients that had high collagen 4 tend to do worse than those that had low collagen 4. We were then able to obtain some pre and post chemotherapy samples from our pathologist collaborator, Dr. Stephen Neighbor at the Tufts Medical School. This is still a small cohort and we're in the process of getting more. But we found that at least in some patients, when we compared the biopsy levels of collagen 4 pretreatment and then the surgery sample levels, which is after this neoadjuvant chemotherapy treatment, we could see that in 3 patients, we could see this very significant increase in collagen 4 abundance, shown here in green, after chemotherapy treatment, suggesting that indeed neoadjuvant chemotherapy can induce collagen 4 abundance in triple-negative breast cancer patients. So collagen 4, as I mentioned earlier, is a basement membrane component. And it is constructed from 6 different chains, and it forms different heterotrimers, and it complexes with laminins to form basement membrane in epithelial tissues and is abundant in breast tissue. We know that in order to disseminate, breast cancer cells need to break down, reorganize, interact with their basement membrane. And collagen 4 is known to interact with a range of receptors such as integrins, CD44, DDR1, and these receptors can activate a range of downstream pathways. But there wasn't as clear evidence as to how particularly collagen 4 was driving migration in triple-negative breast cancer. So we delved into this a little further. And so first we confirmed that by itself, collagen 4 can indeed induce invasion. This is in our 3D spheroid model that I mentioned earlier. And so we compared collagen 1 alone or in combination with collagen 4. Again, collagen 1 is really the most abundant in breast tissue and always there. So we compared those two. And we found that in both mouse and human triple-negative cells, we were able to get a significant increase in invasion when we added collagen 4 compared to collagen 1 alone. And we did a lot of work to delve into the mechanism, and I'm only showing you the data of where we saw differences here. And what we found was that collagen 4 was inducing this effect on invasion via the DDR1 receptor. We could see that the receptor was phosphorylated in response to collagen 4 in these cells, and that this also led to activation of SRC. And we didn't see any effect via integrins or MAP kinase or phospho-AKT of these other pathways. So to then investigate whether this pathway was important for driving the effect that we had seen originally in the decellularized matrices, we took our tumor cells, reseeded them in these paclitaxel treated decellularized matrices, and treated the cells with either a SRC inhibitor or a DDR1 inhibitor. And we can see that inhibiting these pathways does inhibit invasion in these decellularized ECMs obtained from the mice, suggesting that this collagen 4 activated pathway is mediating some of the effect that this paclitaxel ECM is having on the tumor cells. So what I've shown you is that chemotherapy induces changes in the matrix composition in the mammary gland that can promote cell invasion and proliferation, and that these may explain why some patients become resistant and their diseases eventually recur. Collagen 4 is upregulated after paclitaxel and doxorubicin treatment, and this we found increases cell migration via DDR1 and SRC signaling. So the question remains, how does this happen? Who's actually making this matrix? And so a master's student in the lab, Victory, has been investigating this. We know that there's a range of cell types within the mammary gland that can be secreting matrix in these tissues. And so this is just a teaser for what we're looking into in terms of trying to link which cells in the stroma are actually secreting this matrix. Here we've stained human triple negative breast cancer samples with markers for all those cells I showed you previously, but really only see colocalization of collagen 4 with fibroblasts stained with alpha SMA, which is not surprising, but also endothelial cells. We can see some nice collagen 4 around those blood vessels. And so we can actually culture cells of these cell lines in vitro and measure their collagen 4 secretion in the dish. And so we're currently investigating how chemotherapy treatment might impact collagen 4 secretion from these cells in the dish to try and better understand how chemotherapy is leading to matrix production by these stromal cells. And so with that, I'd like to thank the members of my lab. I highlighted those whose work I showed you today, mainly Jackson, Justine, and Victory. I want to thank our collaborators, especially our clinical collaborator, Dr. Stephen Neighbor, for the clinical samples, as well as our funding from various breast cancer foundations, NCI, and the School of Engineering. And thank you for your attention. I'm happy to take questions.
Thanks very much, Madeleine. That was a beautiful presentation. I just want to remind everybody that if you have questions, please post the questions in the Q&A. If we do miss out your questions now, there will be time at the end also to engage both panelists. I guess, Madeleine, what I'll like to do is maybe start off the discussion with, if you can comment on how about collagen 4. So I'm wondering if you can comment on how different ECM molecules might enhance or drive invasion, but from a in terms of the mechanism. So do you think that there's evidence for different mechanistic properties that are driving the different effects of ECM molecules?
Yeah, definitely. I mean, and a lot of matrix proteins will have kind of domains within them that overlap in between different proteins. There can be similarities, and a lot of them can signal through different integrins, for example. But even in work from our lab, the paper I alluded to briefly with the novel method, we focused on collagen 6, which is another collagen. But there, the effects on invasion were driven by an entirely different receptor, NG2, which is a proteoglycan receptor. And that's what was driving the effects. And again, it was not integrin driven, but driven by this receptor, versus here collagen 4, also these effects not integrin driven, and seem to be driven by DDR1. So I think a lot of people will assume that matrix proteins will all signal through integrins, and many of them do. Integrins can also then complex with a range of other receptors like receptor tyrosine kinases to diversify the pathways, but not all of them do, and there are other receptors that can be involved that can mediate these responses. So yeah, you don't always know what's driving it.
Thank you. Okay, questions are popping up. So because you mentioned DDR1, I'll skip to the second question first and then go back. So Deborah and I would like to know, are DDR1 SARC registered in SIN-based analyses of samples from tumor resistance in patients?
Yeah, we haven't looked at that. That's something that would be great to do. We're just in the final steps of kind of teasing out that mechanisms, and that would be good to look at. It can sometimes, in terms of the single cell data, if it's more just gene expression, that doesn't necessarily reflect changes in signaling, but definitely in terms of different targets. So that definitely would be worth something to look into as well.
Okay, Shreya Raghavan asked, what sort of diffusional limit with different ECMs produce differential drug response, especially in 3D?
Yeah, so with those initial experiments I showed where we compared 2D to 3D, we did look at the diffusion, and you can easily do that with doxorubicin because it autofluoresces in the red channel. So with all these different matrices, we could see, for example, how doxorubicin, how much was in the matrix and how much reached the cells. And with those conditions and those different conditions, the different matrices we compared, we didn't see any difference in terms of the fluorescence signal. So I think definitely there can be based on different collagen concentrations, but in the context of that data that we had in our data set and our conditions, we did not see a difference.
Adams asks, what are your thoughts on how this can be targeted clinically? is it feasible to eventually target ECM components?
Yeah, definitely. I think, you know, that's a really exciting new area, both in terms of, obviously there's the receptors that these act on, but also the cells that are making it, the organization in terms of the enzymes that are cross-linking or processing these. So I think there's still a lot of ways that we haven't, that we can use, that we can explore to try and leverage the matrix for targeting that are independent of just, you know, signaling pathways on cells.
Okay, Lester Lambert asks, does integrin-mediated migration necessarily have to rely on upregulation of integrin?
I mean, I guess if it's integrated mediated, then it will depend on integrin signaling, not necessarily upregulation in terms of gene expression. You know, integrins can then be activated, activate proteins at focal adhesions to promote invasion, or crosstalk with RTKs as well to activate other pathways. So not necessarily based on integrin expression, but just integrin activation and engagement in terms of then activating other pathways downstream or crosstalk with receptors.
All right, Stephen Sakuma asks, you discussed it briefly, but do you have any ideas for how to formally test the importance of the 3D conformation branching of ECM?
Yeah, I guess in terms of the in vitro assays, there are different methods that you can use in other labs working on this, on modulating the cross-linking of the matrix without altering the concentration to be able to look at those aspects, for example, and, you know, using other approaches, to modulate the matrix configuration in terms of, forming a matrix that is aligned versus not aligned, or, you know, things like that. So there's definitely like many other labs that are able to generate these different configurations of matrix in vitro. We focused here on more on the composition, and obviously some of these could also be impacting the organization, but not so much changing in that approach. But yeah, that's definitely something that's doable.
Okay, Tony Amelio. Hi, Tony. Says, great talk, Madeleine. Any indication that the chemo treatments are affecting the overall collagen cross-link numbers and or the types of cross-links formed, i.e. labile versus stable?
Yeah, we haven't delved into that, and that's something we're starting to look into. So we will see. Yeah.
Okay, we'll take one more question and then we'll leave the rest for the other discussion. So Zevronai asks, would targeting SARC selectively impact the ECM as opposed to system components, the tumor?
I mean, definitely here the SARC that we're targeting is activated in the tumor cells. So we're targeting the activation of that pathway in tumor cells by the matrix. But would it have other effects? Yes, that's definitely something to look into here. Obviously, we're looking at just the effects on the tumor cells and how that would impact their dissemination. But then targeting this, you would have to consider the other effects on the tumor microenvironment and the tissue as a whole. So yeah, definitely some questions about that.
All right, thank you, Madeleine. So Madeleine will be sticking around after at the end. So if you have more questions, put them into the Q&A, and we'll ask the follow-ups later on. So now we'll present our next speaker. So as Marina gets started, it's my honor to introduce our second speaker, Dr. Marina Pasca di Magliano, who joins us today from Ann Arbor. Marina graduated the University of Naples prior to completing her Ph.D. In immunology at the Institute for Molecular Pathology in Vienna. She trained as a fellow in the lab of Matthias Heybrock at UCSF, where she developed a new area of research in the lab focused on pancreatic cancer. Here she made major contributions to the field by discovering the role of hedgehog signaling in PDAC, Marina started her independent lab in 2008 at the University of Michigan, where she has risen through the ranks and is currently a professor and program co-leader for the cancer cell biology. Her laboratory has made numerous contributions to pancreatic cancer research. She has elucidated cellular signaling mechanisms by the role of oncogenic KRAS in the maintenance of PDAC by developing a novel reversible oncogenic KRAS mouse model called iKRAS. Her laboratory is also making impactful discoveries in the area of immunization of cancer, especially from the perspective of the functionality of myeloid cells and regulatory T cells. On a personal note, I met Marina while I was a postdoc and she was an assistant professor. And I quickly realized that not only is she an accomplished and impactful scientist, but she's an excellent colleague as well. So with that, I welcome Dr. Marina Pasca di Magliano.
Thank you, Cosimo, for the invitation to present and for the nice introduction. I'm going to try to get this right here. Okay. So today I am going to talk, of course, about the cancer microenvironment. This is my contact information for anybody who might want to get in touch later. My lab, as Cosimo explained, studies pancreatic cancer. And one of the reasons that we study this disease is that because it remains one of the deadliest human malignancies. This is a prediction that was published a few years back that shows that pancreatic cancer is expected to become the second cause of cancer death. It is currently the third cause of cancer death in the United States. And even though some progress has been made, the 5-year survival remains, it has barely reached 10% last year. So it was stuck in the single digits for a long time, and it has finally barely made this double digits. For those of you who are not thinking about the pancreas all the time, pancreatic cancer arises from this organ, which is located behind the stomach and the duodenum. And because of the position of the organ, the tumor is rather difficult to diagnose early. The majority of patients present with already metastatic disease. But what is maybe more striking that even those patients who end up being able to get surgery, the vast majority of them will relapse with metastatic disease. So the specific aspect of pancreatic cancer that I've been fascinated by for a long time is the fact that the tumor has this very rich and complex microenvironment. So this slide shows what normal human pancreas looks like on the left. All these cells are acinar cells. They produce and secrete digestive enzymes through a ductal system. You can see here a cross-section of a duct. And the enzymes eventually reach the digestive tract. Of course, the pancreas also has an important endocrine function, but pancreatic cancer arises from the exocrine portion of the organ. Pancreatic cancer, here again, a section of a human tumor, is rather unique because you can see little nests of tumor cells. There is some here and some down here, but the majority of the volume is occupied by components of the stroma, and those include many different cellular types, and of course, a lot of extracellular matrix as well. And Madeleine very kindly sent me this cartoon so I could co-opt it for my talk. So she very nicely showed how important the extracellular matrix is for tumor progression. However, my lab studies the cellular components of the microenvironment. And so this slide, it represents the big questions, the big picture questions that my lab asks. One is, how do components of the microenvironment regulate carcinogenesis? Conversely, how do tumor cells regulate the microenvironment? And finally, we realized that we only had a partial understanding of what the components of the microenvironment were. And for the purpose of today's talk, I'm going to give you 2 short stories that get to 2 of those questions. So first of all, I'm going to take you through some data that we obtained by analyzing human pancreatic cancer using a combination of approaches. And then I'm going to show you a follow-up stories based on some findings that were rather unexpected and based on our multimodal mapping project. So this is just a scheme of how we went about characterizing the components of the human pancreatic cancer microenvironment. So of course we had to start with patient tumors, but we also wanted to have blood correlates. So we got blood samples from the same patient where we had tumor samples. And then we used a combination of approach that included multiplex immunohistochemistry, mass cytometry, and single cell RNA sequencing. And I'm not going to show you all the data today because it will go way beyond my time, and also because I want to show you some unpublished data. But I'll just have to stop for a second to acknowledge that this was a team effort. It was a very large scope project that required a lot of colleagues. It required bioinformatics support and a team of 4 people in my lab who worked together for 2 years to bring this to completion. Together with a larger team of clinicians that were on board to let us have the samples and work with them. So first of all, what were our samples? Our main techniques are mass cytometry and single cell RNA sequencing. And for both of them, we used surgical samples. So you can see here the gray, that's Whipple surgery, which is used for pancreatic cancer. That's in the head of the pancreas, the area proximal to the digestive tract. We also had samples from distal pancreatectomy. We had actually 1 normal pancreas samples and some adjacent non-malignant samples. But what makes our study a little different is that we also had a number of samples that were derived from fine needle biopsy that those were harvested at the time of diagnosis, and they were all from untreated patients. So first of all, I'll just show you a single slide of data from mass cytometry. So this is a technique very similar to flow cytometry, but the difference is that the antibodies are conjugated with heavy metal isotopes, and therefore we can use a lot more antibodies at once. And our panel currently has a little over 30 antibodies. We have a lot of analysis, but the one thing I wanted to bring home here is that while the non-malignant pancreas is relatively homogeneous with most of the cells in this purple category, which is non-immune, our panel has mostly markers for immune cells. So the unknown are actually whatever is not immune. Tumors have a lot of immune cells, but you can also clearly see that there are no 2 tumors that look the same. There is a lot of heterogeneity between patient and patient. We spent most of our time analyzing single cell RNA sequencing data, and we did that with total single cells dissociated from tumors. So we didn't enrich for any specific type of cell because we wanted to understand what was in the tumor and get a sense of how the cells might be communicating with one another. These are UMAPs for our non-malignant samples, as well as 16 pancreatic cancer patient samples. Since then, we now have, even though we were not able to get human samples because of COVID, we have about 22 tumors, and we also have 5 samples of metastatic pancreatic cancer from liver metastasis. But what's reflected here is the cells all together. And so the way this map works is that the cells are clustered and each color represents a different type of cells. And then we go in and give them a name based on their gene expression pattern, as you can see on the right. And for today's talk, I wanted to show you what we found regarding CD8 T cells, because CD8 T cells are not only one of the cells in the body that's responsible for immunity, including anti-tumor immunity, but they're also the target of immunotherapy, which, as many of you probably know, has not been successful in pancreatic cancer. So one of our drivers was understanding why pancreatic cancer doesn't respond to immune therapy. So if we take only those CD8 T cells and we map them, we can now start getting a little more detail, and we get two main clusters. In reality, it's 4 clusters, but 2 and 2 are quite similar. And when we went in and looked at what gene program they're expressing, we could classify them as either effector or exhausted T cells. And that's an example of markers. For example, the effector T cells have higher granzyme B, although you'll see that there is granzyme B expression in the exhausted T cells as well. And then EOMS, which is a marker of T cell exhaustion, is enriched in the what we call the exhausted population. When we compared the relative prevalence of each population in the benign samples versus tumor, you can clearly see that there is a relative decrease in effector T cells and increase in exhausted T cells. At that point, we were interested in knowing what immune checkpoints those cells expressed. And so this is just a cartoon showing some of the immune checkpoint receptor and ligand genes that we looked for. And you can see on the left that when we looked at PD-1 as well as LAG3 and other genes, they were expressed at similar levels in both the effector and the exhausted T cell population. But TIGIT was specifically labeling the exhausted T cells. Then we used a different algorithm which allows us to predict potential interactions. So the way this works is that it maps the expression of receptors and ligands. And if you have a cell with a receptor for a specific ligand, you now have a line. And then you can plot the differential interactions between normal and tumor. So what this plot shows is interactions that are higher in tumor than in non-malignant pancreas. And in this case, we are plotting those interactions where the ligand is expressed by macrophages and the receptors are on different other immune cell types. And one of the couples we pulled up was TIGIT PVR. I'm not showing you, but we pulled this specific couple repeatedly from a whole number of cells in the microenvironment. So we then went back to our site-off panel because we wanted to see whether our RNA sequencing data were reflected by the protein level data. And so you can see here on the left that just similar to what we saw with RNA data, PD-1 and LAG3 were not differently expressed between normal tissue and pancreatic cancer. However, TIGIT was on a range, but it was enriched in cancer samples versus non-malignant pancreas. This is just one example of original site-off plot. So then we went back to the patient's blood because we had collected matched PBMCs from each patient. And we plotted the correlation between expression of TIGIT in the tissue and in the blood. And we found a pretty good correlation indicating that patients that have high TIGIT in their tumor T cells also have elevated TIGIT in their peripheral T cells. And so we become quite interested in validating TIGIT functionally because not only it labels a specific cell population better than other immune checkpoints, and so it might, targeting it might maybe contribute to releasing or reactivating exhausted T cells, but also the correlation between its expression in blood and tumor might allow us to stratify patients for the ones that have elevated TIGIT and where that therapy might be useful. So, of course, while we were doing this in the course of this analysis, we looked at a lot more than CD8 T cells. And one of the populations that we are always interested in are myeloid cells in the tumor, which fall in different groups, as you can see in these graphs, including different groups of macrophages as well as granulocytes and other myeloid cells. So this is a big heat map where each column is a different cell, and the cells are clustered by populations that are similar to one another. And so you can see here, this shows the genes that the algorithm used to generate these clusters. And in particular, there was this group that we call alternatively activated macrophages because of the genes they expressed. And what we noticed was that they had elevated expression of a gene called ApoE, or apolipoprotein E. So what is ApoE? It is actually a very well-studied gene because it's very important for a number of disease processes in human, including cardiovascular disease and Alzheimer's disease. Humans have different variants that confer different risk of Alzheimer's, for example. And more recently, several papers have come out linking it to cancer, mostly in melanoma. So we were intrigued by that, and we decided to investigate the role of ApoE in pancreatic cancer. And this was the work by Sam Kemp, a graduate student in my lab who recently graduated. And it was a collaboration with Howard Crawford, who served as co-mentor for Sam during her time in my lab. So while we were collecting patient samples, we also kept serum from all the patients. So we used the PBMCs for sequencing or site-off, but we collected serum. And so Sam was able to run, or actually Eileen and Sam together ran an ELISA assay. And we were quite surprised because even though the numbers are not huge, there was a quite striking separation between the patients that had high or low ApoE serum levels in terms of survival, with the patients with high ApoE having worse survival. And those are all patients that were included in our study. So they're all patients diagnosed within the last 3 years, but because pancreatic cancer is such a short survival, we already have endpoint for many of those patients, unfortunately. So Sam was intrigued and wanted to know which cells express ApoE. And in addition to macrophages, like I showed you before, there actually was some expression in fibroblasts. And for those of you that are really into fibroblasts, it is the ICAFs, or inflammatory fibroblasts, that express ApoE, and not the fibroblasts that make a lot of collagen and smooth muscle actin. Then Sam went on to validate this by immunostaining. This is now immunostaining for normal pancreas. You can see there is a little bit of ApoE. It is mostly in the blood vessels, but of course there is a lot more ApoE positive cells in the tumor. In human tumors, the same thing, very little staining in the normal pancreas, but a lot of staining in the tumor. And you can see that the staining is quite confined to the stroma and not really present in the tumor cell areas. And again, by single cell sequencing, you can see expression in macrophages. And by co-immunostaining, you can also see that macrophages are also positive for ApoE. You can see here, and there are some fibroblasts that are also positive within the tissue. So at that point, we wanted to know, okay, this is a marker. It's kind of intriguing, but does it have a functional role? And so because this is such a highly studied gene for cardiovascular disease, we were actually able to find a cardiovascular lab that already had ApoE null mice. So Sam did a fairly simple experiment. She took a tumor cell line, which comes from a mouse model of pancreatic cancer, and is derived in pure BLAC6 background, injected them in the pancreas, so either BLAC6 or ApoE knockout mice, also in BLAC6 background. And so reproducible and significant, although not very extensive, reduction in tumor growth. So those tumors are always smaller. They're not gone, obviously, they're always reduced. So that was intriguing. And first of all, she validated that the null mice are indeed null, which is fairly clear. The antibody for ApoE is extremely specific. But then Sam noticed that those tumors also had more cleaved caspase-3 positive cells, indicating that they are a little smaller, and that might be due to increased cell death. She didn't see a change in proliferation, which I'm not showing. She also noticed that there were other changes in those tumors. There were fewer fibroblasts, fewer cells positive for smooth muscle actin. And then she used CyTOF to characterize the immune infiltration in those tumors. So these are just Disney plots showing the different immune populations that she detected. And it is simpler to look at them this way. Each dot here represents a tumor an individual tumor in an individual mouse. And what she consistently found was that there was an increase in T cells, and specifically in CD8 positive T cells. And for those of you who work with pancreatic cancer, you'll know that T cells are fairly rare within this tumor. And so this is an immunostaining to show the same thing. There was an increase in CD8 T cells. At that point, Sam wanted to know whether those increased T cells infiltrating the tumor were active, and they were the reason why the tumors were smaller. So she repeated her transplantation experiment, and she repeated it, but in this case, she also added depletion of T cells. So in a normal wild-type mouse, depleting T cells has no effect on tumor growth, indicating that there is normally no immune response against these tumors. Mice in ApoE knockout animals tumors are a little smaller, and this growth effect is completely rescued when we depleted T cells. So it is definitely driven by a T cell response. So at that point, Sam wanted to know, so how is this happening? And so ApoE has several receptors, but the one that was expressed highly in the tissue was the LDL receptor, which is of course an important receptor for cholesterol signaling or metabolism. And she looked at where this receptor is expressed, and it is on several cell types, but it's highly expressed in epithelial cells, so in tumor cells themselves. So at that point, Sam did a very simple experiment in vitro when she took a pancreatic cancer cell line, added recombinant ApoE or not, and then did RNA sequencing. Those are the genes that were most different between the two. And among those, we noticed that CXCL1 came up as a gene that was upregulated by ApoE. CXCL1 is interesting because there is a rich literature indicating that, including a paper a couple of years ago from Ben Stanger's lab, indicating that it is an important factor in recruiting suppressive myeloid cells and in turn inhibiting infiltration of CD8 T cells. So we thought there might be a link there. So Sam knocked down the LDL receptor, and she showed that if she knocked down the LDL receptor, she partially blocked the induction of ApoE in of CXCL1 in tumor cells that have been treated with ApoE. So you can see here, this is the knockdown. It's only about halfway. For some reason, we haven't been able to fully knock it down. But if you treat tumor cells with ApoE, you upregulate expression of CXCL1, and that's partially reduced when we knock down LDL receptor. So it's clearly not the only thing that's happening, but it could be one link. And the very final piece of this is that Sam wanted to know how is ApoE connected to CXCL1 expression. So when she analyzed her RNA sequencing data of tumor cells that had been treated with ApoE, one of the pathways that was increased was NF-kappa-B. NF-kappa-B signaling is complex, but the take-home message is that if the signaling is inactive, P65 is sequestered in the cytoplasm, but when the signaling is activated, P65 translocates to the nucleus. So Sam took tumor cells either control or treated with recombinant ApoE. And you can see that treatment with ApoE increases the green nuclei. So those are nuclei where P65 in green has translocated to the nucleus, and that's quantified here. At that point, the hypothesis became that ApoE activates NF-kappa B signaling, and NF-kappa B signaling in turn activates transcription of CXCL1. And to test this possibility, she pre-treated tumor cells with an NF-kappa B inhibitor and then treated them with a recombinant ApoE. So if you take tumor cells and treat them with recombinant ApoE, there is a very prominent upregulation of CXCL1. And with the addition of the NF-kappa B inhibitor, there is less upregulation. However, you'll notice that compared to the basal level, there is still upregulation. So there is probably a role for NF-kappa B, but it's definitely not the whole story. So what she found is that ApoE promotes immune suppression by promoting the expression of CXCL1 by tumor cells. We would like to understand what are the other links between ApoE and CXCL1. And even though I only talked about CXCL1 for the sake of time, there is actually a whole family of CXCL cytokines that's upregulated in tumor cells treated with ApoE. We also would like to take a therapeutic approach and combine ApoE with other immune regulating agents. And finally, we are breeding mice to have a genetic model which is both a model of pancreatic cancer, but in the context of loss of ApoE. And finally, this is my last slide, concluding today's talk. So we are really interested in the microenvironment in pancreatic cancer, with the realization that it is very heterogeneous, and that's definitely an area that will require more study, because it is likely that not all patients have the same microenvironment, and that's something that will be important for therapy. However, some common themes have emerged, one of which is that TIGIT is upregulated in pancreatic cancer, and it specifically correlates with exhausted T cells. Macrophages that enter pancreatic cancer have elevated ApoE expression. They are quite different from macrophages in the healthy pancreas. And in ApoE expression contributes to immune suppression in part by upregulating CXCL1. With that, I need to end by thanking everybody in my lab. This is only half the lab because we are still on 50% shifts, so you can only get half the people at any given time. A lot of collaborators, those are all people that are part of our pancreatic cancer group at the University of Michigan, and or most of them. And also, of course, funding by NCI and the American Cancer Society. And with that, I'll be happy to take any questions.
Thank you very much, Marina, for a beautiful presentation. Just remind everybody to please put in your questions in the Q&A. And while those people are putting in their questions, I'll just ask a kind of a general question. So from what I understand, so you do have systemic ApoE going on. And so I'm wondering how that might interplay with cholesterol metabolism, because as you know, in P53 mutant, there's very sensitive to cholesterol. And so do you think that there's an interplay here between cholesterol and what you're observing? And the follow-up to that would be, is there a therapeutic opportunity for statins with your ApoE, or is that conflicting?
Yeah, no, I mean, I think, so to the interplay with cholesterol, I think that's very possible and very likely, and it's something we actually would like to look into using, the samples we already have and prospectively collecting samples from patients. And then I think the role or the potential for statins in cancer is really interesting. There is some correlative evidence that people on statins have a lower risk of certain cancers. So there's definitely something that could be happening there. And, you know, those are some of the questions that we would like to address. Actually, Howard and I are starting to work on a grant. So there are definitely more questions than we have answers.
Thank you. Okay. Guy Salvinson asks, is there a correlation between ApoE isotype and pancreatic cancer in humans?
That is aim 3 we're working on. We don't know because we are trying to see whether, we are trying to see whether we can get that data for the patients for which we already have, you know, other data. But that's a really interesting question. So in mice, you can't address that because mice only have one ApoE version, but in humans, that's a really interesting question. I think there are at least two related questions. One is whether having a certain variant. So for melanoma, some of the variants have a higher correlation with the risk of developing melanoma. So there is definitely precedent for that, but people haven't looked for pancreatic cancer. And then the other question is whether having a certain variant predicts worse outcome or better outcome. And we don't know. I guess I don't have very many answers, but we have lots of questions.
Okay, AJ Dixit asks, is there a difference between ApoE levels in residential versus resident versus recruited macrophages?
Yeah, so we did some pseudotype and other types of analysis to figure out which macrophages elevate ApoE. And I think the fair thing to say is that within the tumors, there is both happening. So the resident pancreas macrophages have some level of ApoE, and then the bigger population seems to be a monocyte-derived macrophage population that has high ApoE. We also observed upregulation of ApoE in blood monocytes of pancreatic cancer patients. So we think that the bigger population is probably monocyte-derived, but the rest of the macrophages also will express it.
So Marina, in your early, you were noting about the heterogeneity between patients and the stromal cells. I'm curious, did you what they're classifying as antigen-presenting CAFs? And also, did you notice heterogeneity between patients in like the ratios of the different CAF subtypes?
So we do find a group of antigen-presenting CAFs or mesothelial cells, depending how you like to call them. So they're definitely there. And in terms of the ratio, that's a little bit of a difficult question to answer with single-cell sequencing data. We don't like to use that data to quantify different cell types. The reason for that is that when you dissociate the tissue, different types of cells react differently, and you artificially enrich for certain kind versus others. So what we had in our study, which I didn't show the data of, is multiplex immunohistochemistry. And so we mostly relied on that to look at the relative, with some supporting data from CyTOF, to look at the relative number. The problem is that while it's very easy to look for MyCAFs because they have such high smooth muscle actin, it's fairly challenging to really look at the number of ICAFs or APCAFs, in a tissue-based way.
Okay, thank you. So we're going to open up the discussion. We're going to open up the discussion. So Madeleine, if you could share your excellent, thank you. And we will go back to some questions for you, Madeleine, but Devronai has a question for Marina. The mild degree of tumor growth inhibition in the ApoE knockout mice may suggest opposing function for ApoE in other TME tissues or cell types. Was this evaluated?
Not yet, but it is, I completely agree that is, and that's something we see in other cases too. For example, with hedgehog signaling, which is something we've had a long interest in, what we have seen is that when we inhibit that, you have a positive effect on the fibroblasts, but a pro-tumor effect on immune cells, for example. So that is something that we are starting to look at. So even though the sort of canonical receptor for ApoE is the LDL receptor, there are other less canonical receptors that have been described. And one of them is expressed in T cells. So that is something we're interested in to look at, but that's sort of along the lines of what we're thinking. There is something, the other thing we have tried is combination therapy with anti-PD-1, and that had no additional effect in shrinking the tumors. So there's something else that's preventing those T cells from becoming fully activated.
Okay, Madeleine, there's a question from Sevi Sarika. Is it feasible to modulate doxorubicin diffusion rate with your specific drug?
Yeah, I guess I'm not sure what you're saying about with your specific drug. I mean, I guess probably you're talking about the diffusion in terms of the matrix, and I think kind of relates back to one of the other questions about how matrix structure can be used to alter diffusion, and I think that's definitely something that is worth delving into, and that other labs that can create assays where the matrix has very different organizations and structures could use that. But I think in general the matrix can in some contexts and will impact drug diffusion, so that's definitely a part of it. It was just in those particular contexts that we did, that was not the limiting factor, but it was a very small range of conditions. So I think that's an important factor to keep in mind.
Okay, so it looks like we've answered all the questions in the Q&A. So I want to thank you both, Madeleine and Marina, for 2 excellent presentations today. It was really fun to get to hear about your work, and thank you for sharing some unpublished data as well. So that will end the presentation portion. So if you, the panelists, could please stick around. There will be the trainee discussion, and thank you everybody for joining us today.